I will come to the final topic discussion, cytotoxic T cell. Cytotoxic T cell, cell mediated immunity, no doubt in that. Now my question is whether cytotoxic T cell requires antigen presenting cell or free antigen is okay. You please pause the video and answer. It requires antigen presenting cell. If it is not going to require antigen presenting cell means it is humoral immunity. The free antigen will be there, your B cell will take that. So here it is T cell, so it requires antigen presenting cell. I am going to draw that APC. This is T cell receptor. This is cytotoxic. You try to answer. What could be this antigen presenting cell? Antigen presenting cell could be anything. It even could be macrophage, it could be B cell, it could be all types of cells except RBC. Now what is this? You have to answer. Already we have gone in depth regarding cell mediated immunity. Yeah, this is MHC. Now you tell me, this is MHC 1 or MHC 2? We already explained for helper T cell H2 or 2H. So 2. So MHC 2 is for helper T cell. So this is MHC 1, very very important. Now again CD will come here. Yes, what CD? Yeah, for MHC, for T helper cell, it is MHC 2 and CD4. I told it is a combo. And what combo is going to be here? CD, which is going to be present on the surface of cytotoxic CD8. Note it down. And same function. What will be the function of CD8? Yeah, CD8 will check. Yeah, this is antigen, this is T cell receptor. Yeah, all this check is done. CD8 has checked. C1 is coming from antigen presenting cell. Yeah, the antigen is being taken here, or MHC is bringing the antigen. So the function of MHC is to bring the antigen to the cytotoxic T cell, or cytotoxic T cell goes in search of the antigen which is containing MHC. Now imagine, yeah, this is free antigen. Just concentrate. This is free antigen. What will cytotoxic T cell do? It will not, because T cells property is it will take only antigen coming with MHC, and antigen coming with MHC is given from antigen presenting cell. That is why the name is antigen presenting cell. Even T cell can help cytotoxic T cell, that is helper T cell. So helper T cell also can help cytotoxic T cell. Now coming to function of cytotoxic T cells, obviously killing. That is why it is also called as killer T cells. So if they ask which is also called as killer T cells, the answer is cytotoxic T cells, not helper T cells. Now next question, how cytotoxic T cells, cytotoxic, cyto is cell, toxic is toxic. How cytotoxic T cells is going to kill the antigen? Another thing I want you to tell, difference between CD4, CD8. CD4 is MHC2 helper, CD8 is MHC1 cytotoxic. And what I want you to understand is this MHC1 usually takes endogenous antigen, or for example this free antigen, instead of this free antigen the entire virus itself will be there. This is virus which contains antigen. So remember, what I want you to mention is MHC1 usually the antigen will be present within the antigen presenting cell. It will not come from outside and processed. What I want to mean is usually MHC2 will contain processed antigen coming inside. Like for example here antigen is there, this is macrophage, this antigen is thrown out. So either this can be taken by B cell or this antigen will enter into the antigen presenting cell, and this antigen is usually MHC2 and given to helper T cell. This diagram, now actually what I am saying is very important, though the video is lengthy, just concentrate here. The antigen is being thrown. This is now free antigen. So free antigen can be taken by B cell. Free antigen will be taken only by B cell. But if this antigen enters into the antigen presenting cell and being presented along with MHC2, now this antigen, yeah, this can be taken by helper T cell. I want to tell the difference between MHC2 and MHC1. MHC2 is exogenous antigen. That's what I want you to tell. Not dictum, just understand. For MHC1, the antigen is present within the antigen presenting cell, or this antigen presenting cell is, for example, macrophage, and this macrophage is taking the entire virus inside. The virus itself is inside, and that virus is broken down into antigen. So the antigen is being broken down within the cell, endogenous. That's what I have written. Endogenous is for CD8 MHC1 cytotoxic T cell. For this antigen is coming from outside, but once it is entered inside, it is not free, but the antigen was broken down outside. That is why it is called exogenous. So note it down. CD4 MHC2 exogenous, MHC2, MHC1 endogenous, and MHC1 cytotoxic T cell. Yes, cytotoxic T cell is MHC1 and MHC1 is endogenous. And this helper T cell MHC2 CD4 is exogenous antigen. So this two you remember. How cytotoxic T cell going to kill the antigen? Mainly three mechanisms. One is perforin here. So this is antigen presenting cell. You can tell MHC1, very good. This is that antigen. This is TCR, T cell receptor. This is T cell cytotoxic. So this will be CD8. So now what happens? This is cytotoxic T cells. Once it binds with the antigen, perforins, endogenous perforins are going to be released from the T cell. Second step is it can release granzyme or lysozymal enzyme. Zyme means breakdown. Lysozymal enzymes or granzymes. Third step is apoptosis. So that once this combines, the cytotoxic T cells can kill this antigen along with the antigen presenting cell by the process of apoptosis. That also you can. If they ask how the cytotoxic T cells is going to kill the antigen, by releasing perforins. Perforins means what will happen? This is going to release the perforins. This may even break the cell membrane. The cell which is presenting also can be going for death. In case of cytotoxic T cells releasing perforins, it can also release breaking down enzymes called granulozyme or granzyme. Granzyme, and it can also kill by releasing tumor necrosis factor alpha. Note it down. Tumor necrosis factor alpha can also be released by cytotoxic T cells, which can cause apoptosis. Apoptosis is programmed cell death. Here, for example, if this is infected, this is antigen or this is cancer cell. This is infected cancer cell which is having that virus, and the antigen is being presented by MHC1. These perforins are being released. This entire cell also can be destroyed by the cytotoxic T cell. So that's the power of this cytotoxic T cell. So it can either destroy the antigen or it can even destroy the antigen presenting cell as a whole if that is going to be a damaged T cell, and that destroys by a process of apoptosis. And with this we will complete today's video on cell mediated immunity. So I started with types of immunity, then I told why it is cell mediated, why it is humoral, fluids, free antigens can flow freely in the blood, can flow freely in any fluid, that will be taken by B cells. T cells takes only processed antigen coming from MHC or coming from antigen presenting cell, and I told the difference between T cells, helper T cells, cytotoxic T cells. I discussed about primary immune response, secondary immune response because of memory T cells, and I also explained about how the antigen is activated, how the antigen is presented, how the antigen is processed, how the antigen is activated, and how helper T cell communicates with antigen presenting cell, how proliferation takes place, various interleukins, communications, types of helper T cells, Th1, Th2, Th17, all those things we discussed, and then finally how cytotoxic T cell is going to destroy the antigen. It could be either the antigen alone coming with MHC, sometimes the cytotoxic T cell can also destroy the antigen presenting cell if the cell itself is infected. That is what I was telling. Cytotoxic T cell usually acts against endogenous antigens which is present within the infected cell, and helper T cell usually fights against exogenous antigen. The antigen is outside, but it comes into the antigen presenting cell. If it is not at all coming to the antigen presenting cell, that antigen will be taken by B cells producing antibodies. So that's what I want you to understand the difference. Hope this video is very useful to you, and if you like this video, please share to our friends, and if you have not subscribed to our channel Physiology Lectures by Dr. Senthil Kumar, please subscribe to our channel, and you can even put in a comment box what chapters you want for further discussion. Any doubts in this also you can ask in the comment section, and I will be very happy to post your doubts and queries in our comment section. Thank you. We will meet in the next video. Thank you. Happy learning.